Module 3, Hair and Nails. Hair and nails are two offshoots of the epidermis, so it's important to study both of them. Hair is made of tightly packed epithelial cells, so it is considered epithelial tissue, and humans have this hair tissue covering almost all of their body. A hair is divided up into two parts, the shaft and the root. The shaft is the part of the hair that extends above the skin's surface. The root is the portion of the hair in the skin, ending in the hair bulb. Most of the hair itself is made of keratinized cells, arranged in 3 concentric layers. Before I talk about the 3 layers of hair, I first want to discuss the 2 types of keratin, hard and soft. Hard keratin contains more sulfur than soft keratin. The sulfur is what makes the keratin harder. Skin cells are made of cells that have soft keratin, but most hair cells are made with hard keratin. That's why burning hair smells so badly. If you've ever accidentally scorched the hairs on your arm, perhaps while making yummy s'mores over a campfire, you've smelled the sulfur in the hard keratin, combining with hydrogen to chemically produce hydrogen sulfide, the same gas that rotten eggs produce. Well, let's get back to the cross-section of a hair. The innermost layer of hair is the medulla. It is made of 2 to 3 layers of dead cells with the soft keratin. Very fine hair has no medulla, but coarse hair usually has medulla. The next layer is the cortex. It makes up most of the hair thickness. The cortex contains several layers of cells with hard keratin. The outermost layer is the cuticle, and it is made of a single layer of dead cells with hard keratin. But the cells in the cuticle are layered like shingles on a roof, overlapping each other to provide an extra measure of protection. The hair grows out of the hair follicle. Remember that even though the follicle descends down into the dermis, it is made mostly of epithelial tissue. There are 2 regions of the follicle. The internal epithelial root sheath attaches the hair root to the follicle. The external epithelial root sheath is white tissue that sometimes is attached to a hair if it is pulled out with a bulb of white tissue attached to it. And the whole hair follicle is surrounded by a dermal root sheath. In this diagram, you can see the blood vessel extending upwards, creating a dermal papilla. It extends up into the hair bulb. Now, right on top of that is the matrix. This is a mass of undifferentiated cells, meaning they have not specialized. As the matrix produces more cells via mitosis, they will begin to differentiate into special cells so that they can become part of one of the hair regions, medulla, cortex, or cuticle. Then those cells will keratinize to form hard or soft keratin, depending on what type of hair layer they are part of. If the matrix of a hair is destroyed, the hair will no longer grow. Why is it when we go to get haircuts, we don't have to have the hair on our arms or eyebrows cut, only the hair on our head? Well, some places of your body, hair can grow long, and other places it cannot. Hair grows about a ½ inch each month. That means about 6 inches a year. And there are times when the cells of the matrix of hair follicles undergo mitosis rapidly. Those are called growth stages. But there are also times when the matrix cells do not undergo mitosis. Those are rest stages. In a growth stage, cells are being made, differentiate, and keratinize. Thus, the hair will grow. During a rest stage, the follicle shrinks a bit, tightly holding the hair in place. Then at the end of the rest stage, the old hair is pushed out of the follicle by a new hair growing behind it. That's why sometimes you find lots of hair in your hairbrush, but you're not going bald. As those hairs are lost, new ones are coming in right behind them, at least when you were young. Have you ever wondered why you need to get regular haircuts, but you never have to cut the hair of your eyebrows or eyelashes? Why don't they grow as long? Well, it's a good thing they don't, or we couldn't see very well. But let's talk about why this is. It turns out that different hair follicles in the body have different growth and rest stages. The hair on your head, for example, may have a growth stage that lasts years, so your hair can get very long. Then the follicle may have a rest stage for 1 year after that. Yet while that follicle is in a rest stage, the follicles around it are likely in a growth stage. In fact, at any point in time, up to about 90% of the hair follicles on your head are in a growth stage. And as some hair follicles move from rest stage to growth stage, you lose some of your hair, about 100 hairs a day. Yet you don't go bald because you have tens of thousands of hairs on your head. Now let's talk about eyelash hairs. Eyelash hairs have a growth stage of about a month and a rest stage of a little over 3 months. That means that eyelash hair only gets to about a ½ inch in length before it hits the rest stage. So now you can see why different hair lengths on the body are a result of the different lengths of growth and rest stages in the hair follicles. Take a look at this figure again. At the base of the hair bulb are melanocytes. Remember, they are cells that produce melanin, a protein pigment. These cells have those extensions called processes that are filled with melanin. Matrix cells of the hair engage in phagocytosis to engulf the melanin-filled processes. That's how hair becomes colored. I already mentioned that there are two types of melanin, dark eumelanin and red pheomelanin. Well, color does not come from how many melanocytes there are because everyone pretty much has the same concentration of them. It results from how much melanin the cells take in. If the cells take in a lot of eumelanin and not much pheomelanin, then the hair is dark in color. If the cells take in lots of pheomelanin and not much eumelanin, then the hair is red. Blonde hair is a result of the cells not taking in much of either pigment. If blonde hair has a little pheomelanin, then the hair is a golden blonde. If the blonde hair has a little eumelanin, then the hair is an ashy blonde. And if the cells don't take in any melanin, then the hair is white. As people age, their hair turns gray as a result of the melanocytes producing less and less melanin. That means that the hair cells cannot take any in. Now let's look at nails. Nails are very much like hair in structure. If you look at a nail from its side, you can see that it is like a hair shaft. The part of the nail you can see lies above the skin surface. The nail root is under the skin, and there's a nail matrix at the bottom of the root. Just like the hair matrix, the nail matrix is the place where undifferentiated cells undergo mitosis. Then they specialize and keratinize with hard keratin. That's how a nail grows. But unlike hair matrices, a nail matrix doesn't have a rest stage. Instead, it grows continuously. Nails just grow more slowly than hair. The nail matrix does extend a bit out of the skin. That little white moon-shaped part at the base of your nail is called the lunula. The lunula is the upper part of the nail matrix and is thicker than the rest of the nail. That's why it's whitish. It's more difficult to see the pink skin tissue below it. As long as the matrix is alive, nails can grow. Nails are very helpful to us. Not only can they remedy an itch when we have one, they also give us protection on the ends of our fingers and toes. When we grasp something, especially with our fingertips, the nails provide structural reinforcement so we can better hold things. Module 3, the Integumentary and Skeletal Systems, Experiment 3.2, a closer look at follicles. We're going to take a closer look at hair follicles in this lab. You have a slide that's human skin with hairs, and that's the one that you want. I've been saying don't get that one in the previous labs. This is the time we want to use this one because we want to take a closer look at the hair follicles. So go ahead, set your slide under the microscope at 40 power and focus in, and you're wanting to look for the wavy line that marks the epidermis, and you should be able to see several teardrop-shaped hair follicles. So I'm going to put that under my scope, center, and focus, and you'll see some oblong shapes underneath the dermis or within the dermis, and those are the hair follicles we're talking about. Look amongst those and see if you can find one that's really well defined, that the bulb itself is kind of circular, because the cross-section kind of comes at random spots, and we're trying to find one that got the cross-section pretty close to where the bulb is rounded. And you want to see if you can find some cross-section that has at least a little bit of white in the middle. And if you can see a hair sticking out of the follicle, that's really ideal. So you may have to go from one to the other to find all of these parts as we talk about them. Once you find your bulb, go ahead and increase the magnification to 100. So I'm going to do that right now and center that and focus it again. All right, so I'm focusing on one that's got a nice white section in the middle. And once you've done that, you should begin to see at this power some individual cells, again with the nuclei stained a little bit darker inside of those cells. And the divisions at this point between the hair, the medulla, the cortex, the cuticle should be visible as well to you. You should be able to, using the diagrams in your text, identify a little bit better what you're looking at, as well as the follicle. Now the follicle has internal epithelial root sheath, and it also has an external epithelial root sheath. You also can see the dermal epithelial root sheath, or dermal root sheath as well. Take some time looking at this one. If you can't see that on one of your follicles you're looking at, move to another one. And again, using your diagrams that are in the textbook as your guide to figure out what you're looking at. Then once you've done that, let's move on to the next step. You want to focus on that rounded bulb follicle that you can find, and we're going to focus on 400 power with that one. So I'm going to move mine over to the rounded bulb and 400 power now. And focus, recenter it a little bit. There we go. You should see something even more in detail now. Again, the cells should be very noticeable at this point. Looking at the bottom of the bulb is where we want to start. That bottom section is the matrix. And notice the shape of the cells in that matrix. You can tell the extra density in that region, and there's just more of them packed in, just more packed in than the rest of the hair follicle. Just above the bulb now, let's take a look at the regions, the dermis below the follicle and the regions above the follicle. Look at the line of tissue surrounding the outer layer of the follicle. Okay, the outer layer of the follicle, the line. That's called the dermal sheath. There's internal and external epithelial root sheaths that will be pretty much the same color, but that's the dermal sheath. So you may have a hard time identifying those two, but again, using your diagrams to help you navigate through this slide will be very helpful, as well as looking at the slides that I'm showing you. But the external sheath is a little bit wider, and the internal sheath is made up of smaller cells. Now the hair itself should be distinct. If you have opportunity to have a slide that's got a little hair coming out of that where you can see it, it's going to be very different from the follicle. The tissue should be a little darker, brownish colored, a little more dense looking, or light's not passing through it as well. And that may be one opportunity for you to be able to tell where the internal epithelial root sheath is. Now take a look at the hair even closer. And the layer of tissue right outside the hair then will be the internal epithelial root sheath. Okay, at this point, go ahead and draw that whole bulb and the part of the follicle that you're looking at in the under the microscope, labeling what you can identify and what power that you're observing it under. And then now we want to look more closely at the hair shaft itself. So you're going to adjust your slide a little bit more, looking at the hair shaft, and hopefully you'll be able to see even more detail what that hair looks like. It may be very difficult for you to identify the difference between the medulla and the cortex of the hair, but you can see the cuticle if you adjust the fine focus. Remember, we don't like to use coarse focus at 400 power, but using the fine focus, move it up and down so it goes in and out of focus. You're kind of looking below and above the hair follicle itself, and you might be able to see a yellowish outline around the hair. If you see that yellowish outline, you should see hopefully some darker lines that outline that yellow outline. And you might be able to see them overlapping like shingles. That's what we're going for. If you can find that, you have been able to observe the cuticle of the hair. And so if you can see that under your microscope, go ahead and make an illustration of that in your notebook, identifying those parts and the power that you're looking at your slide underneath. When you've completed that, then go ahead and clean up everything and put it all away.